welcome to Mr. Go's Biolectures. This Biolectures is very important to a student who has just joined medics and a student who has just joined advanced level. Make sure you like, subscribe, like and share so that you can see more of the videos. So without wasting time, last time we are supposed to look at something called glycolysis. And the other biolecture we saw the mitochondria. Now this one we want to look at the glycolysis here. It can also be called the cell metabolism, also called the cell breakdown or the glucose breakdown. We are going to look at how glucose is going to be broken down from the 6 carbon atom to the 3 carbon atom, which is called the pyruvate. Without wasting time, let's begin our biolectures. Now we are beginning with something very interesting here. I want us to pay attention and we see here. I'm having what to call, this is the muscle tissue, which is this one here. Hope you have this. Now this muscle tissue here, of course we have cells there. And then I'm going to just cut a bit of it. I've cut like this. And now I'm going to do what? I'm going to zoom and look at the tissues there. We have the cells which are here. I see these cells here. This is a cell, a muscle 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 cell here. This is a muscle fiber. And therefore what I'm going to do here, and we are seeing something that is in red here. This one that we are seeing in the red here. This one is a blood vessel. What's the name of the blood vessel? Remember, if you can go back, we see that the blood vessel which is trying to enrich the materials, that is towards the fore limb, so what to call the subclavian artery. You can say this is supposed to be what to call the subclavian artery. Something very interesting. This is subclavian artery. The one you are seeing here, this one here, that you see in the green here, subclavian artery. So when it reaches here, it is going to just branch into what to call the tibial and the ulnar and then the tibial artery. This is one of this one, what to call the ulnar and then the tibial artery. But we don't need this. Our major focus is here. Now when I extrapolate this one, when I extract this one here, when I extract, I've extracted this region here containing one cell. It is the one we are seeing here. Is what I've extracted from here. It is containing what to call this blood vessel I've talked of. This one, which cell is what to call the subclavian what? Artery. Subclavian artery, which is enriching the forelimb. Now, this subclavian artery here, of course, it is containing what to call, this is blood. This is blood. This is blood. Blood tissue. Of course, the blood tissue, this is what to call the blood tissue. So this blood tissue here is consisting of what to call nutrients and like amino acids, very many nutrients there, the ions and so on. But our major focus is supposed to be now the glucose. This is the glucose which is nutrient here. This is the glucose that we're having here. The glucose. And this glucose here, it is 6 carbon atom. So this is number 1, this is 2, this is 3, 4, 5, 6. So glucose is a 6 carbon atom. Now we want to see how glucose is going to be broken down. But before we see how glucose is going to be broken down so we can get energy from it, we have to first see how is glucose moved from the blood vessels into the tissues. How does glucose reach into my cells, the muscles here, which we are seeing here. Now, of course, this is the this subclavian artery here. It goes and branches into tiny blood capillaries. The blood capillaries, the tiny ones. So I want to call this is going to find what to call this. I want to call the tiny what? Blood capillaries. What's going to happen here? Of course, glucose can leave the blood capillaries by diffusion because where it is here, the blood tissue, in the blood here, it is highly concentrated. So it can easily diffuse from the region where it is in high, region where it is low, either by passive or by fast-state diffusion. It can be by fast-state what? Diffusion. Now, what to see here, the question is, how does glucose cross? Because this is our cell we are seeing here. But how does glucose cross into what to call the what? The cell. Something very interesting here. On the cell membrane of all cells, we have what to call certain transporters. These transporters here have what to call the gates. These are called the gates, what we see here. Or these are gates. These are called the gates. Then we have these are the gates here. Then we have what to call the receptors. But how do you call this Ugandan here? This person here who's having the receptor, the same time what to call the gate here. These are called the GLAT transporters. They are called the GLAT transporters. The G, this one here, is called the GLAT transporter. Members, we must appreciate that these GLAT transporters here are in various muscle cells. Okay, some muscles and also other body organs like the liver. The ones in the liver are not the ones that are the what? The muscles. For example, let's look at something interesting here. We have something very interesting here. I'm just going to use this. We have what to call those that are found. Like we have what to call now the blood. That's one. We have what to call, of course, in the brain when we talk about the blood, we talk about the red blood cells. Then we have secondary. We have what to call the blood brain. It's supposed to be the blood brain barrier. Is that membrane which is separating the brain from the blood. This one here. Then also have what to call this is the brain, the blood. And also have what to call this one is here. This one major here. They are also what they are to call the gut. The gut one. These are called the gut one transporters. You get the gut one transporter. These ones are just located in what to call the blood cells. And also what to call the heme. That is can be also the heme that is in the foetus. This can be the baby. The foetus. That membrane of the foetus is having what to call the gut one transporters. So we also have something very interesting here. We have what to call the kidney. This is phase 1. I'm calling this one phase 1. We have phase 2 here, which is what to call the gut 2 transporters. We have what to call the kidney. That is something interesting. We have one. We also have what to call we have what to call the liver. We have what to call kidney. Have what to call the liver. Those are the major ones. This one kidney and then the liver. We have to call the gut to what? Transporters. Then also the pancreas. The pancreas can also be here. So the pancreas, the liver, and the kidney, they have their channels. Their transporters are called the GLAT what? GLAT, not GLAT. GLAT to what? Kidney transporters. Then we have part 3 here. We have what to call now. We have these are called the GLAT 3 transporters. Here we majorly have what to call that's what we call the placenta. This is between the mother and the baby. Then we have what to call also the kidney can also have these transporters, can have both GLAT and also what to call the GLAT 2 and 3. Then we also have what to call the kidney, then what to call these are mentioned here, and also the neurons. The neurons have this one here. Then we have what to call lastly category 4. We have what to call the GLAT. The GLAT 4 transporters. Here we have what to call the muscle. These ones are the muscles. They also what to call in the fat adipose, that is the fats or adipose tissue. So why am I bringing this one here? It is just you to know that specific type of cells have what to call their transporters, so to not confuse them. Now let's get back there. Now we have seen that what is the GLUT for this is the muscle cell. That means it's supposed to be the what? The GLUT 4 transporter. So that means it's supposed to be the what? This is supposed to be the GLUT 4 transporter. Now let's look at this one here. Now the glucose has entered into the cell through the gates here, and then we don't want to beginning now the process. When the glucose enters into the cell by whichever mechanism, because it has to first bind, after binding here, then these pores are going to open. When opening, glucose flows into the what? The cells. But we have to prevent this glucose from leaving the cell. What's going to happen here? We shall have what to call, we are going to add a phosphate to the glucose. So this one is a glucose molecule. We're going to add the phosphate. This is going to be, I'm going to label it here like this. This is number 1, has carbon number 1, 2, 3, 4, 5, 6. So what's going to happen is that this phosphate is going to bind on what to call 6 on the 60th carbon, glucose carbon, on the 6th carbon atom. That means it is going to bind here. This is the phosphate ion. So it has bound onto the what to call the 6th carbon atom. Something interesting here. Now what is going to form here? This is supposed to be, what is the name of this? This is called now, what's the name of this compound here, or the molecule here? This is supposed to be called what? Since the glucose, the phosphate is on 6, supposed to be called 6. That is the this glucose. This is glucose. Supposed to be called glucose 6 phosphate. So that's what we call the glucose 6 phosphate. Now, this glucose 6 phosphate, we have to see what has bound this phosphate here. It is supposed to be what to call an enzyme, because there has to be an enzyme. Which enzyme is this? Now we have what to call 2 enzymes. We have what to call the hexokinase. This is one. Then we have what to call the glucokinase. This can also be called hexokinase 2. This can also be called hexokinase 4. Hexokinase and then glucokinase. Something very interesting here. Now, this hexokinase, it is just specifically in all, it is in all cells, all body cells, apart from what we call the liver cells. Apart from the what? Apart from the liver cells. So this glucokinase is majorly in what we call the liver only. It is not in any other cell apart from the what? From the liver. So when glucose is taking place in the liver, the enzyme which is going to bind the phosphate onto this glucose, the form called glucose 6 phosphate, is what we call the glucokinase. So it is going to depend on what I'm talking of here. But why is the ATP, where has the phosphate come from? This phosphate has come from what we call the ATP molecule, which has donated what we call the phosphate, and then remains what we call ADP. You get? Plus inorganic phosphate, which has bound onto this carbon number 6 here. Let's continue. That's something very interesting. Let's move on. Something very interesting. Let's move on. Now we have this. So after getting what we call the glucose 6 phosphate, what is going to happen? This glucose 6 phosphate is going to undergo what we call isomerism, the arrangement. This one, the glucose 6 phosphate, is like an aldehyde, but we want to convert it to what we call the ketone. You get? So the hydrogen atoms, the carbon number of atoms, and oxygen atoms are the same. But what we're going to have is just arrangement of the what? Of the bonds. So what's going to happen is that when you isomerize, you form what we call the fructose 6 phosphate. It's supposed to be the fructose 6 phosphate. Now, which enzyme was rearranged this, isomerized this? Supposed to be the hexose isomerase enzyme. This is the enzyme that has done that. From what to call this one here. Now the fructose 6 phosphate is going to undergo what to call is going to be converted into this compound here. What is this one here? This compound here, excuse me, is going to add it is going to gain some phosphate, which is added here on the carbon number 1. So after adding this, it is going to have what to call a given compound, which is going to be what to call the fructose. Fructose 1,6 bis 4 bisphosphate. We're saying the C1 because the phosphate is on carbon number 1 and also on 6, but it is bis. There's a difference between the bisphosphate called the phosphate biphosphate. So we have what to call bisphosphate, bisphosphate, and what to call the biphosphate. So this one here, if the carbon, this is now, let me say this is the glucose molecule. Now, if the carbon atoms are far away from each other, are not the same number of, are not, if the phosphate groups are not on the same carbon atoms here, it is called bis. For example, one is here, another one is here. This is called bisphosphate. But if the phosphate groups, one is here, another one is here, they are just next to each other, this is called what? Biphosphate. Now, which enzyme is going to involve here? Something very interesting. Something named phosphor. Supposed to be the phosphor. Supposed to be, that is converting this to this one here. Supposed to be the fructose. Okay, the phosphofructose. Supposed to be the phosphofructose. Phosphofructose kinase type 1 enzyme. Can you say phosphofructose kinase enzyme? For me, what to call this one here, fructose 1,6 bisphosphate. So this glucose, this fructose 1,6 bisphosphate here is going to undergo what to call further breakdown. And what has happened here? There's something that is very interesting here. Because seeing that a phosphate has been combined here, where has it come from? A phosphate here, it has come from what to call the ATP, breaking into what to call this ADP, then giving what to call the phosphate, which came and combined on what? On this one here, the phosphate, inorganic phosphate, which has combined here. Now, that means you have so far used how many ATP? So far 2. This is the first ATP, this is the second what? ATP. Now, this fructose 1,6 bisphosphate is going to break down into 2 compounds, which are 3 carbons. So this is going to break in the middle like this. So this one is going to go, this one also going to go here. So we shall have this one. This is 1, 2, 3. So this one is called now the dihydroxide. This is what called dihydroxide acetone phosphate. Sometimes you normally abbreviate it as DHAP. DHAP, that's what we call the hydroxide acetone phosphate. Then this one is going to be called the glycerol 3 phosphate. Glyceraldehyde 3 phosphate. Also abbreviated to call the glyceraldehyde 3 phosphate, something like that. So we have a phosphate which is here, glyceraldehyde 3 phosphate, or GA3P. That is the phosphate group is here. Now what's going to happen is that there is a given electron carrier here, which is called the NADH, also called the reduced NAD. What's going to happen is that this reduced NAD here, it is going to react with this the GA3P here, And then what's going to do is going to dehydrogenate, remove the hydride ions. So the hydride ions are going to be removed, because when you talk about the hydride just a combination of the hydrogen ions and also what to call what? The electrons. So what's going to happen is that this Gl3p here is going to lose the hydride ions, and then are going to be accepted what to call the oxidized NAD to form what to call what? The NADH or reduced NAD. But remember, as I told you earlier, that this dihydroxyacetone phosphate, DHAD, this DHAD here, I told you that DHAD, I told you that it converts into what we call the what? The Gl3p. What's going to happen is that since this has been converted into another format of what we call the Gl3p, we are going to be having the 2 Gl3p, the 2 glyceraldehyde 3-phosphate, meaning these are 2 of them. You get the point. So what's going to happen is that since there are 2 of them, the Gl3p here, we shall have what to call 2 NAD plus, and also the 2 NADH. So this one not only go dehydrogenation here, we also undergo what we call some kind of reduction, which is called now phosphorylation. The phosphate is going to be added on what? On carbon number 3. This is on carbon number 1. Okay, it's okay. Number 3 here, so what we call a phosphate is going to be added here. So we shall have something like this. The phosphate has been added here, and then this phosphate has been added here. So what's going to happen is that we are going to, how is this phosphate going to be added here? This is going to come from what we call the ATP, and then from what we call the ADP, plus this phosphate which has been added on the what? On the 3rd carbon atom here. So this reaction is catalyzed by the enzyme. What's the name of this enzyme here? So the enzyme here, which is working on this reaction here, is what we call the glyceraldehyde 3-phosphate dehydrogenase. Is what we call glyceraldehyde, so-called the glyceraldehyde. Glyceraldehyde 3-phosphate dehydrogenase enzyme. Something very interesting. So what's going to happen is that what we're going to form here, remember we have phosphorylated. This is supposed to go this side here, because this one is glyceraldehyde here. This is supposed to go on this one here, not here, because there's the phosphate has been added onto this one here. Admin this one form, the phosphate is going to be here. Sorry about this. So it is going to be here, that is the phosphate. So, and then the one that was here, this is the phosphate or the carbon number 1. So this is now the compound which are going to be having what we call the 1 3 base 4. 1, where we saying 1 because the phosphate is on carbon number 1. The 1 comma, those are the 3, then the base phosphor, that is glycerate. That's what we call the glycerate. The 1 3 base 4 glycerate is what is going to be formed here. Now, what's going to happen that this 1 3 base 4 glycerate, we have seen the enzyme there, is going to lose what we call one of the phosphate on carbon number 1. So this phosphate is going to be lost here, the phosphate that is here. There's a difference between a phosphor and what we call what? I think I explained this phosphor and then what we call biphosphate. So that means this is going to be called what? Bisphosphate. Why? There's a carbon 1 is here, another one is 3 here. So there is another one that is in between the two. They are not so adjacent to each other. So what's going to be is that this 1 3 the phosphoglycerate is going to lose a phosphate. This phosphate here is going to be accepted what to call the ADP. That is that means you have what to call, this is it, is going to be accepted by ADP plus the organic phosphate. forming what we call the first ATP molecule directly. This is the first ATP molecule. You have to know that. The first ATP molecule. You get? What's going to happen is that which enzyme has been behind this physiology here? This is what we call the phosphoglycerate kinase enzyme. How like that? Something very interesting. It's supposed to be called the phosphoglycerate kinase. That is phosphoglycerate kinase enzyme. That is the enzyme which is behind that. Then that means that what are we going to form here? We shall form an organic, so we shall form what we call a molecule which is remaining with only the phosphate on the carbon number 3. So we shall have what we call only this phosphate remaining here on carbon number 3. The one carbon number 1 has gone. And then we are forming this compound here. What's the name of this compound here? Since it is remaining with only 3 carbon atoms, so it is going to be called the 3-phosphoglycerate. That is, I'm going to put it this side here. It is 3-phosphoglycerate. So this 3-phosphoglycerate is going to undergo what to call some kind of rearrangement, like a form of anerobic. What is going to happen here? The carbon number, the phosphate on carbon number 3 is going to shift to the carbon number 2. So the one that is here, this phosphate here, is going to shift and come to what? To the second carbon atom. And then we shall form a given structure. That means we say the carbon is at 2. So we say 2. This is going to be called what? This is going to be 2-phosphoglycerate. Why? Because the carbon atom number, because the phosphate is on what? Carbon number 2. The phosphor supposed to be the 2-phosphoglycerate. Which enzyme is behind here? The enzyme is very interesting. The enzyme is none other than the phosphoglycerate mutase enzyme. Something very interesting. I'm going to put the enzyme here. This enzyme behind this, behind the game here, is what we call the phosphor. That is phosphoglycerate. That is mutase enzyme. That is something very interesting here. So that's the enzyme which is going to convert what we call is going to isomerically arrange this from the 2, the phosphate from the 3 up to the 2 carbon atom. So what's interesting here? Now this phosphoglycerate, the 2-phosphoglycerate is going to make some kind of a phenol, whereby the phosphate is going to attach on, is going to substitute the hydrogen ions on what we call the phenol. So as we can form something called the phosphenol. You get phosphenol, a phosphate, but it is on a given group on the phenol. That is, that means we are going to form something like this. What I'm trying to mean is this: if I'm having what we call, this is my kind of, let me say this is my phenol here, and then there is an OH here. What is going to happen here is that the phosphate is going to substitute this one here, remove the hydrogen, and then it attaches there to form what we call the P. You get? That means it has been attached here. You get? So we are going to form what we call the phosphenol pyruvate. Something very interesting. It's supposed to be called, this is called what? Phosphenol pyruvate. The phosphor. We are still saying phosphor because the phosphate group is there. Phosphenol. That is pyruvate. Sometimes it can also be abbreviated as the PEP. Now this, which enzyme has been this one here? Something very interesting here. The enzyme is none other than what we call the arenase enzyme. That's something very interesting. It's called arenase enzyme. Then this one now is a 3-carbon. All of them are 3-carbon compounds, but the difference is that it is still having the phosphate on it. So what I'm going to do, we are going to remove that phosphate. Are we going to do it? We are using what to call a given enzyme, which is going to remove this one here. What's the name of this enzyme here? Supposed to be the pyruvic or pyruvate kinase enzyme. That is mean the P kinase. That means the P kinase enzyme is going to, the pyruvate kinase enzyme is going to do what? Is going to remove is going to remove the phosphate. But if this phosphate is removed, what's going to happen here? The phosphate that being removed, it is going to attach onto this is the phosphate. This is ADP. So it is going to attach what to call the what? This ADP here, form what to call what? ATP. Therefore, we are forming the second ATP molecule directly. So the first molecule was somewhere here. This is the second ATP molecule. The first ATP molecule was around here. This one here was the first ATP molecule. That means you have so far more manufactured the two. But remember, these glycerol 3 phosphate were 2. That means you have how many ATP? 2 ATP molecules. Also 2 ADP. Even here we shall have how many? We shall have what to call 2 ADP and 2 ATP. We shall also have this compound. We shall have this one here. The 1, 3, this one, there are 2. There are 2 of them. And also this one here, there are also 2. And then also this one here, they're also going to be how many? They're going to be also 2. And also this compound here, they're also going to be 2, the PEP. Then also the pyruvate, you're going to form what called the pyruvate, which are also 2. This one called the what? The pyruvate. Or can also be called what called the pyruvic acid. What? Pyruvic acid, something very interesting. To look and we see, we have got what called the pyruvic acid here. So this pyruvic acid here, we have this arrow here. It is showing us that we have anaerobic and also what's called aerobic here. This pyruvic acid here, in absence of oxygen, when there is no oxygen, what is going to happen? This pyruvic acid here is going to be converted into what we call the lactic acid. Lactic acid. What's going to be behind here? We have what we call the NADH2. You get the NADH2 is going to donate its hydrogen ions or its hydrides. You get? We have what we call the NAD. I'm going to put it here. The NADH2. Something very interesting. The NADH. So this NADH here, what is it going to do? It's going to add what we call, it's going to react with pyruvic acid here. After reacting, what's going to happen? It is going to give it what we call the hydrogen what? Or the hydrides. Then what's going to happen? It is going to remain as what we call the NAD plus. In other words, also called oxidized what? NAD. Now what's going to happen here? When you add hydrides on this one here, we shall form what called the lactic what? Lactic acid. But which enzyme is behind this physiology here? There's an enzyme that is going to allow the binding of these hydrogen ions from this up to this one here. This enzyme is what we call the lactate dehydrogenase. Lactate dehydrogenase enzyme. So this lactate dehydrogenase enzyme here is very important. It converts what we call the pyruvic acid to what we call the lactic acid. But this lactic acid is very poisonous, it's very toxic. What happens is that it goes into the liver. It goes into the liver through the blood vessels. It goes into the liver. Now reaching in the liver, what's going to happen here is that this lactic acid is going to be converted into what we call that is glucose. That's one. It can also be what we call to manufacture called ATP. That means that we have got what we call the ATP. Also, the energy has been got to what we call lactic acid. Then lastly is going to be that, remember, this lactic acid is very toxic. It is very toxic in the body. So what's going to happen is that when it becomes too much, it lowers the pH, and then this can damage the cells. That's why if you look at the clinical relevancy of this lactic acid here is that, or the clinical correlation is that, when you test an individual or someone, and this person is having a lot of lactate dehydrogenase enzyme, it means that this person here could be having one of the tissues damaged. It could be the liver, it could be the kidney, it could be the heart. Why am I saying this? Because this lactate dehydrogenase enzyme is mostly contained in what called the cytoplasm of those organs that are very sensitive with the body. That means when you see it within the blood, a lot of it, like for example 1 week, or even after 1 week, it is still there, the levels are still high. It means that there is an organ which got destroyed. and then the contents in the cytoplasm got split and then poured into what we call the what? The blood. Therefore, you get what we call damaged organ. Also, the brain could be affected. Could be having anemia. The person could be having anemia. Could be having cancer. All those are what we call effects of what we call, if someone have what we call lactate dehydrogenase. But if it is normal, during exercise, it is okay. It can come because it has to convert the pyruvate into lactic acid. It can also take back this lactic acid here into what we call the pyruvate. This one reaction is reversible. It can take it back. Another one is we can convert lactic acid into what we call the pyruvate. That is if it becomes too much in the blood, and that we don't want that toxicity. That is it. Then what about now in aerobic respiration when there is oxygen? When there is oxygen, what happens is that this pyruvate or the pyruvic acid here is going to first be decarboxylated. That's something very interesting. It is going to be converted into what we call the acetyl coenzyme A. That is called A. This one is a 2-carbon compound. This acetyl carbon A is then going to go into what we call the Krebs cycle. The Krebs cycle is going to come out to call the oxaloacetate A, which is a 4-carbon plus the 2-carbon, which is this one here. Then we shall continue with the cycle until we form back again. This is what we call the Krebs cycle. So during this process here, we're using what we call energy. So if oxygen is present, we just have to convert that into acetyl coenzyme A and then fill to the Krebs cycle and get more energy. You get? That is something very interesting. Now let us look at now the energy yield. Something very interesting here. Energy yield. Remember, there was 2 ATP molecules. That means before that I can just give some brief summary. Just give a summary. I think this is the summary. Let me use here. So in this summary here, we had that what was the where was the process taking place here? It was what we call the cytoplasm. That's something very interesting. Glycolysis takes place in the what? The cytoplasm here. Then what is the substrate? What was the disparate substrate that was being broken down? It was glucose. I mean, this is glucose that was broken down. Then what is the product that we gave out? This was what we call the what? The pyruvate. Also called the pyruvic what? Pyruvic acid. So what is now going to happen in this pyruvate during aerobic respiration? This is going to give us what we call the lactic what? Lactic acid. This lactic acid is toxic, so it is going to be converted into what we call glucose. Can also be converted into what we call ATP, and then some can be converted back into what we call the what? The pyruvic what? Acid. So that it can go during oxygen, it can go back in the Krebs cycle to give more energy. Then what about now during aerobic respiration? Aerobic respiration here. What's going to happen is that the pyruvic or pyruvic acid is going to be converted into acetyl-CoA, which is going to be fed what we call the Krebs what? Krebs cycle, and then more energy is gotten from there. Then what are the energy acceptors? Another one is the energy carriers here. We have what we call the NAD H. These were how many? These were 2. And remember, each NAD yields how many? 3 ATP molecules. That's something you have to note down. Then we got how many? We had how many? 2 ATP molecules. Since there are 2, that means there are going to be how many? There are going to be 4 ATP because there are 2 glycol and 3 phosphates. These are 4 ATP. That means this is going to give us 3 ATP. That means since there are 2, they're going to become 6 what? ATP indirectly. So when you get these 4 plus 6, they get how many? Total ATP, total gross is how many? Is it 10 ATP. But then you remove the 2 that we put in, because there was the one that we put here during phosphorylation here. This first one here. Also there was another phosphorylation somewhere whereby we put in an ATP. This was around here. The second one here. We don't count the one for this one here on the glycol and the 3 phosphate. So we get how many? 10 ATP. Then the 10 minus the 2, that means you get how many? 8 ATP. The net, this is the net ATP. That means during glycolysis, we get the net amount of ATP is 8, and then the net is 8 ATP. You get? So don't forget, in summary, we said that glycolysis is almost like 10 steps mechanism whereby the glucose molecules enter into the cells via what? Via what we call the glut. transporters. Then it is going to be phosphorylated to form what we call the glucose 6 phosphate, which is going to undergo what we call several reactions, whereby the glucose 6 phosphate is going to undergo what we call isomerism to form what we call the fructose 6 phosphate. So the fructose 6 phosphate is going to undergo what we call certain kind of breakdown, whereby you're going to form what we call the fructose 1,3 bisphosphate, which is going to break down going to form what we call the dihydroxystone phosphate. Another one is going to form what we call the glycolysis 3 phosphate. So the dihydroxystone phosphate is going to be converted into what we call the GA33 or the glycolysis 3 phosphate, because this one does not undergo further breakdown. So the guys using which enzyme? The triose phosphate isomerase enzyme. So this glycolysis 3 phosphate is going to undergo several mechanisms breakdown until we form what we call the pyruvate, which is the 3 carbon atom. Then during oxygen, the pyruvate is going to be converted into what we call stercoenzyme. Then when there is no oxygen, the pyruvate is going to be converted into what we call lactic acid, which is going to go to the liver and then detoxify to get energy and also to get glucose and so on. How many gross ATP? There are how many are 10? Net 8. Thank you very much for watching. This is once again Mr. Saija. Thank you for watching. You don't have to cram these ones here. It's just a matter of understanding the flow, the mechanism. Thank you. God bless you. Bye bye.